Imagine for a second that you're trying to crack a combination lock. Just a standard three-digit lock. It's got, 1,000 possible combinations.
Right, which takes a while, but you can do it.
Exactly. If you sit there long enough, you can brute force your way in.
Right.
But now, imagine a lock that doesn't just have 1,000 combinations, but actively changes its own code 10 billion times a day.
Oh, wow. Yeah, that changes things.
Right. Like every single time you figure out a number, the lock has already mutated to a different one.
And that right there is the exact mathematical nightmare medical researchers faced in the 1980s. I mean, the target was HIV, and the numbers were entirely, completely in the virus's favor.
Welcome to this deep dive. For you, the insanely curious learner, today's topic is basically a masterclass in how a single medical breakthrough changed the world. We're looking at a stack of sources all focused on pharmacology, specifically antiretrovirals or ARVs, for managing HIV.
Which is such a massive topic.
It is. So our mission for this deep dive is to understand the incredible math behind how these drugs suppress this incredibly fast mutating virus, why combination therapy is literally the whole point, and how these molecules shifted from this desperate toxic intervention to a global prevention tool.
It's a huge journey.
Okay, let's unpack this. Because we're looking at a trajectory where a diagnosis went from a near-certain death sentence in the 80s to something that today, with reliable access to a daily pill, means a completely normal lifespan.
The sheer scale of that shift is, it's unprecedented in modern medicine.
Totally.
But to really appreciate how far we've come, we have to look at pharmacology's very first desperate swing at the virus and why it ultimately failed.
And to understand that first attempt, we need to understand the lock itself. So, looking at the scientific foundation in our sources, there was a massive scientific turning point in 1970, which is wild because that was years before HIV was even identified.
Yeah, that was the discovery of reverse transcriptase. Two scientists, Howard Temin and David Baltimore, they independently figured out that certain viruses had this really unique enzyme.
Right.
And it completely shattered the core assumption of biology at the time, which researchers called the central dogma.
The central dogma being the idea that genetic information is a strictly one-way street, right? Like it goes from DNA to RNA to protein. You don't ever go backward.
Precisely. But retroviruses, which HIV is, they carry their genetic blueprint as RNA. So when they invade a host cell, they deploy this reverse transcriptase enzyme to essentially reverse engineer their RNA into DNA.
Which is like reading a translated book and writing it back into the original language on the fly.
Exactly. And once it's in a DNA format, they can sneak it directly into the host cell's genome. It was such a radical discovery. It won a Nobel Prize in 1975.
So fast forward to like 1983, 1984. HIV is identified as the cause of AIDS. And scientists immediately realize this reverse engineering process is the virus's glaring vulnerability.
Because humans don't use reverse transcriptase.
Right. So if you can poison that specific enzyme, you can theoretically stop the virus without killing the host's normal cellular function.
That was the hope, yeah. But you have to remember the desperation of the era. They didn't have time to design a bespoke drug from scratch. People were dying by the thousands. So researchers at the National Cancer Institute just started screening massive libraries of existing compounds.
Throwing things at the wall to see what sticks.
Literally looking for anything that would jam that enzyme.
Which brings us to the fascinating and honestly tragic origin of the first ARV, which was AZT, or zidovudine.
Yeah, the backstory here is wild.
It wasn't even built for HIV. It was synthesized back in 1964 by Jerome Horwitz as a potential leukemia treatment.
Right, a cancer drug.
And it completely failed. It was just sitting on a shelf collecting dust in over 2 decades.
Until they dropped it into a petri dish with HIV, and incredibly, it worked. It blocked reverse transcriptase. So Burroughs Wellcome, the pharmaceutical company, they pushed it into clinical trials immediately.
Like incredibly fast.
Yeah. Oh, yeah. The FDA approved it in March 1987, which was just 25 months after the first human trial began.
25 months is unheard of. But, okay, let's look at the reality of prescribing AZT back then. Was it basically like trying to put out a forest fire with a single highly pressurized garden hose?
That is a very apt analogy. The dosing was essentially carpet bombing the body. Patients were taking 1,500 milligrams a day.
Wow.
And the sources describe severe side effects, massive bone marrow suppression, profound anemia. People needed frequent blood transfusions just to survive the treatment itself.
Just to survive the drug. But that gets into how the definition of success is completely shifted, right?
Yeah. Early on, success just meant a patient surviving a few extra months. It meant seeing a brief transient bump in their CD4 count.
And just to clarify, CD4s are the immune cells that HIV specifically targets and destroys.
Exactly. So the toxicity of AZT was horrific, but the alternative was certain death. It was a genuine emergency. But scientifically, the single drug approach was, well, naive about viral evolution.
Because that garden hose didn't just fail to put out the fire. It mathematically guaranteed the fire would mutate and get smarter.
It did. It forced the virus to become significantly more dangerous.
Which brings us to the most crucial turning point in HIV history.
The pivot to heart.
Right, and the shock that sparked that pivot came in 1993 with the Concorde trial. This was a huge study in the UK and France, and it absolutely stunned the medical field.
Why? What did they find?
They found that early AZT monotherapy, so using only that one single drug, had absolutely no long-term survival benefit. None.
But the virus just bounced back anyway.
Every single time. And the reason comes down to that math we were talking about. By 1995, mathematical modeling by David Ho and George Shaw revealed that HIV was replicating at a speed no one had even imagined.
Right, the numbers in the source material are staggering.
An untreated patient was producing 10 billion new virions every single day.
10 billion. Wait, so if the virus is making 10 billion new virions a day, it's like if you buy 10 billion lottery tickets a day, you are mathematically guaranteed to win the jackpot. That is exactly how it works. And what makes it a guaranteed win is that reverse transcriptase has no proofreading ability.
Oh, unlike human cells.
Right, human cells catch their typos. HIV doesn't. It produces a mutation in almost every single replication cycle.
So it's constantly churning out random variations.
Right.
And the sources give a really clear example of this with another early drug, lemivudine.
The M184V mutation, yeah.
Just one tiny typo, a single amino acid substitution. And if the virus happens to make that specific typo, it easily escapes lemivudine. The drug just becomes useless.
Exactly. Because you are actively killing off all the vulnerable viruses with the drug, but you're leaving behind that one mutated version, so it just takes over the entire population.
You're just clearing the playing field for the worst version of the virus.
You've created a perfect evolutionary pressure cooker. And understanding this math is what birthed highly active antiretroviral therapy, or HAART, at the 1996 Vancouver conference.
Which earned David Ho Times Person of the Year, right?
It did, because it fundamentally changed the approach.
So what does this all mean for you listening? The first principles takeaway here is that combination therapy, which means taking at least 3 active drugs from 2 different classes, it isn't just this vague, more is better intuition.
No, not at all.
It is an explicit exploitation of combinatorial probability, like the chances of a virus randomly mutating to resist 3 completely unrelated drugs simultaneously is practically impossible.
You multiply the probabilities, and the odds go from inevitable to, well, virtually 0. You force the virus into a mathematical corner it can't mutate its way out of.
So if combination therapy relies on hitting the virus from multiple angles simultaneously, how exactly are these drugs doing the hitting inside the body?
That brings us to the specific choke points.
Right. So if the virus is an illegal factory, how do these drugs shut down the assembly line? Can you walk us through the major classes?
Definitely. The sources detail 6 major choke points. Let's start at the door. The first class is entry or fusion inhibitors. They literally stop the virus from entering the cell entirely. They jam the lock.
Which leads to this absolutely fascinating real-world proof of concept, the story of Timothy Ray Brown, the Berlin patient.
Oh, this is an incredible case. What's fascinating here is how it validates the entry inhibitor logic. In 2007, Brown needed a bone marrow transplant for leukemia, and his doctor deliberately found a donor with a rare genetic mutation called CCR5 delta 32.
And that mutation basically means the host's cells are missing the specific doorknob that HIV uses to get inside.
Right. It effectively locked the doors to his cells. His new immune system rejected the virus, and his HIV went into remission for 13 years, completely off ARVs.
Yeah, obviously we need to clarify for the listener that a bone marrow transplant isn't a scalable cure. It's incredibly dangerous. But it was an incredible validation that if you lock the door, the virus fails.
Exactly. But for most people, we rely on drugs that work after the virus gets inside. So the second choke point involves NRTIs, like AZT or today, tenofovir.
The faulty building blocks.
Yes. As the viral enzyme tries to copy its RNA into DNA, it grabs one of these fake building blocks, it clicks into place, and bam, it terminates the DNA chain.
The assembly line just halts.
Right. Then you have the third class, and NRTIs, like efavirenz. Instead of being a fake block, they actually bind to the reverse transcriptase enzyme itself and jam its motion physically.
So you're throwing bad bricks onto the belt and throwing a wrench in the gears of the belt itself.
Precisely. Now, if the virus gets past both of those and finishes its DNA, we hit the fourth choke point, integrase inhibitors, or NSTIs, like dolutegravir.
What do those do?
They block the virus from integrating that rogue DNA into the host's actual chromosome. This class is the anchor of most modern regimens now because it has a really high barrier to resistance and very low side effects.
Okay, so that's 4. What if the virus is already integrated in producing viral proteins?
That's the 5th class, protease inhibitors, or PIs, drugs like ritonavir. This was the class that actually made HAART possible back in 1995. They block the cleavage of proteins.
Like breaking the chemical scissors.
Yes. The cell still makes new viruses, but because the proteins aren't cut properly, the virions are completely defective and non-infectious.
Incredible. And there's a 6th class mentioned, though they aren't technically ARVs themselves.
Right, PK boosters, like cogasistat.
Yeah.
They don't attack the virus, they just inhibit your liver enzymes to keep the companion ARV drugs circulating in your blood longer.
So once researchers prove they can use this assembly line wrecking crew to lock the virus out or shut it down, they asked a really revolutionary question. Could we use these same drugs to protect people who don't have the virus?
Which completely flipped the script. Because over time, the definition of success for ART shifted from just keep a patient alive to driving their viral load down to undetectable.
And viral load is the lab measurement of HIV RNA copies in the blood, right?
Yes, and undetectable means getting it under 20 to 50 copies per milliliter.
And that leads to one of the most important concepts today, UU. Undetectable equals untransmittable.
The clinical data for this is just rock solid. The PARTNER and HPTN 052 trials proved this. Across thousands of couple years of follow-up where one partner was positive and undetectable and the other was negative, there were 0 linked transmissions.
0. That is amazing. So treatment itself is prevention.
It is.
But here's where it gets really interesting. This brings us to pre-CAP, pre-exposure prophylaxis. It uses the exact same molecules developed for treatment, but gives them to HIV-negative individuals.
Right, specifically Truvada, which is a combination of 2 NRTIs, tenofovir, and emtricitabine.
Yeah, I want you to clear something up for us, because there's a big misconception that pre-PP is either an absolute 100% magical force field, or that it's totally useless based on some early headlines.
Yeah, you're referring to the 2010 iPreX trial.
Exactly what happened there.
So in that trial, the overall top-line efficacy was reported as 44%. Which sounds terrible for a prevention pill.
Yeah, 44% sounds basically like a coin toss.
But that number included everyone who was handed the pills, whether they took them or not. When researchers looked at participants who actually had detectable levels of the drug in their blood, the efficacy was over 90%.
Wow, okay.
Efficacy is entirely dependent on adherence. The drugs work phenomenally well, but you have to actually take them.
Which shows that translating the science into human behavior isn't always smooth. And translating it into public policy hasn't been without tragedy either.
No, it hasn't.
Looking at the debates and denials in the history of ARVs, there was this massive 20-year debate in the medical community about timing. Hit the virus early or wait?
Because early AZT monotherapy failed so badly, doctors initially utilized this watchful waiting strategy. They wouldn't prescribe ARVs until a patient's CD4 count visibly declined.
But that debate finally ended in 2015, right?
Yes, with the START trial. It proved that starting ART immediately, regardless of how high the CD4 count is definitively the superior strategy.
So clinical hesitancy was one thing, but state-level denial is a completely different chapter. And I want to explicitly state to you listening right now that we are impartially reporting the contents of the source material here. We aren't taking any political sides or endorsing viewpoints, just relaying the historical facts documented in the research.
Right, and the sources detail the tragic episode of AIDS denialism in South Africa between 1999 and 2008.
Under President Thabo Mbeki's government.
Yes. His government actively restricted public access to ARVs based on arguments that questioned whether HIV even caused AIDS.
And the cost of that policy was devastating. The sources cite a 2008 peer-reviewed analysis by Chigwedere and colleagues. They estimated that this specific policy delay resulted in over 330,000 excess deaths.
Over 330,000 lives. It's a stark reminder of what happens when public policy actively rejects pharmacological consensus.
It really is. Now, looking away from the past and toward the next frontier, the current live debate in the field is about long-acting injectables versus daily pills.
Yes. like the cabotegravir and rilpivirine injection approved in 2021. Instead of a daily pill, you get an injection every month or two. It's incredibly convenient.
But you mentioned something earlier about the pharmacologic tail issue. What is that?
So if a patient takes a daily pill and then stops, the drug leaves their system pretty quickly. But with a long-acting injection, if they stop coming in for their shots, the drug lingers in their body at low levels for a long time. That slow tapering off is the tail.
And why is that bad?
Because lingering low drug levels don't fully suppress the virus. So if they're re-exposed or their own virus rebounds, it's the perfect training ground for the virus to develop resistant mutations.
It's that pressure cooker all over again.
Exactly.
Which leads to one final big clarification. If the viral load is undetectable, and these drugs are this good at shutting down the assembly line, why isn't a cure? Why can't people just stop taking the pills once it's undetectable?
That is the holy grail question, and it comes down to the latent reservoir. The latent reservoir. Right, so HIV integrates its DNA into your CD4 cells. Most of those cells are active, and the drugs protect them or they die off. But a small fraction of CD4 cells are resting memory cells.
Oh, they just go dormant.
Yes. They carry the viral DNA, but they are not actively replicating. And since ARVs only target active viral machinery, the copying, the cutting, they can't touch these dormant cells. The virus is hiding in plain sight.
So the second you stop taking the drugs,
the mathematical pressure is gone. Eventually, a resting cell wakes up, the factory turns back on, and the reservoir reactivates.
Wow. So to synthesize this whole journey for you listening, we've gone from the rushed, incredibly toxic single drug days of the 80s to the mathematical revelation of HAART to today's single manageable daily pills.
And the impact has been staggering. Just a year or two after HAART was introduced in 1996, AIDS deaths in the U.S. dropped by 40 to 50%.
Which is just incredible. And today it forms the foundation of UNAIDS's 95-95-95 global targets.
If we connect this to the bigger picture, ARVs are really a rare medical marvel. They act simultaneously as chronic life-saving therapy and primary prevention.
They really are. And it leaves me with one final kind of provocative thought for you to ponder on your own. The mathematical modeling and those combinatorial strategies that finally cornered this fast-mutating virus, they were born out of absolute desperation.
Complete desperation.
But what if this exact playbook, forcing a virus into a mathematical corner where it cannot simultaneously mutate against multiple choke points, what if that is the secret to defeating other highly adaptable threats?
Oh, that's an interesting angle.
Right. Could the brutal lessons we learned from HIV resistance be the blueprint we eventually use to conquer rapid onset superbugs? Or, who knows, even figure out a cure for the common cold?
The math is definitely universal.
It's something to think about. Thank you so much for joining us on this deep dive. Keep questioning, keep exploring, and we will catch you next time.